Many of you may know 10, 12 years back, we had very few treatment options. I very well remember when I was a resident at LSU, and I was being mentored by a lot of my, like Dr. Woltering, Wang, Ramirez, and others. And we had very few treatment options to offer. And when it came to PRRT, I profoundly remember we used to send patients to Europe or had to turn them to some clinical trial. It wasn't available. And fast forward to 2018, we had first FDA approval for PRRT for use of neuroendocrine tumor in form of lutetium-177 dotatate or Lutathera. So today we will discuss what is that and what is on the horizon. Before we discuss about Lutathera, those who are new to the field, let's kind of prime them with the concept of theranostics and how these radiopharmaceutical or targeted radiotherapies work. So PRRT is a highly targeted and effective form of radiation. Okay. Our traditional or conventional radiation is you sit in front of a machine and we zoom a beam of radiation, target towards a particular cancer tumor, and it works pretty well. But as you know, in advanced disease, when the cancers unfortunately metastasize, it's not just located in one organ. It's spread around even throughout liver or bones or several areas, big and small. So you can't really radiate the whole entire body because there's a lot of normal intervening tissues. So you're going to give a lot of side effects. So PRRT is a critical solution to that. We are able to administer the radiation, but specifically to the tumor cell, avoiding the normal tissue. So basically made the drugs which are radioactive. How does it work? So think of neuroendocrine tumors. I'm using this new software, so it kind of got rid of the animation I had. So I'll try to just explain you. Might have to imagine a little bit here, okay? So this lock represents somatostatin receptors. These somatostatin receptors are present in abundance in neuroendocrine tumor patients, especially the well-differentiated, especially the grade 1 and grade 2, and some grade 3. So if this receptor, we treat them like a lock. What if we have a key? You know of a key, somatostatin analog, these drugs like sandostatin and lanreotide. These are somatostatin analogs. When we put them in contact with somatostatin receptor, they bind just like a key will bind to a lock. So that's a good solution. What if we make this key radioactive? This is a symbol for radioactivity. So we tag lutetium-177, which is a radionuclide. It's a heavy metal which emits radiation. So, and inject that drug into your body. So that drug will find wherever the somatostatin receptors are, bind to it, and since it's radioactive, is going to take that radiation along with it to the tumor tissue and then radiate it. That's the concept of theranostics. Now, this concept, is being used for somatostatin receptors in neuroendocrine tumor, PSMA for prostate cancer, and several other tumor types, because the concept is same. All you need to do is find the target and get a new key which binds to that target. For today's talk, we will focus on our target, which is somatostatin receptor, and our key, which is lutetium-177 dotatate. So here, this is a GEP-NET gastroenteropancreatic neuroendocrine tumor cell here, which on its surface have somatostatin receptors, SSTR2 specifically. And this drug, the dotatate part is the key that binds to that receptor and is tagged to this lutetium-177, that radionuclide, which is emitting this radiation. So when you inject this drug, it binds to the receptor on the surface of the cell, gets internalized, and when the drug is inside the tumor cell, it's emitting this radiation. And what is this radiation doing? How is it killing the cancer? By causing the breaks in the DNA. You see this DNA is being spliced. That's how radiation kills anything living. That's why radiation is lethal. So it kills the DNA of the cells this damaged DNA, the tumors with damaged DNA cannot replicate, cannot divide, cannot thrive, and it dies off. That's the mechanism of action. That's how this drug works. So this was the concept, but do we know it works in our patients? Yes, Europeans and some other countries have been using lutetium-177 dotatate in neuroendocrine tumor patients for a really long period of time. But in the US, we had access to this because we finally had a randomized phase 3 study called NETTER-1 and proved it conclusively in a prospective manner that yes, this class of drug works. So what was this study about? The midgut neuroendocrine tumor patients, the small bowel neuroendocrine tumor patients with metastatic disease who had progressed on other lines of therapies, they were randomized. Half of them got Lutathera and other half got higher dose of Sandostatin. And what we found is, this is the Kaplan-Meier curve of progression-free survival in NETTER-1. The green line represent the patients who got Lutathera, and the gray line represent the control arm, the ones who got higher dose of Sandostatin. These patients have been followed up for several years now, and now we know, after follow-up, the median PFS, or the average amount of time for what disease was stable in patients who received Lutathera, somewhere around 28 months, roughly, as compared to around about 8 months. A huge difference. And this was pretty profound. And based on this data, FDA approved this drug for use in metastatic gastroenteropancreatic neuroendocrine tumor patients in the United States, those who have progressive disease. So it's a big win. We no longer had to send patients elsewhere. Patients could get treatments in the US. Fast forward to 2025, there are over 200 places in the US administering Lutathera almost every day to hundreds of patients. It's been a big game changer of a treatment. And not only this treatment works so well to stabilize the disease for so long, but it also helps with quality of life. Many anti-cancer drugs might be very effective in controlling the cancer, but comes at a cost of giving side effects, additional toxicity. So when we studied the quality of life in NETTER-1 patients, the quality of life metrics on Lutathera arm is depicted by the purple line, and green line is the control arm. You see, the patient reported better quality of life, those who were treated with Lutathera. So not only the drug helps patients had better median PFS, but also improved the quality of life. Those are our two biggest goals when we treat metastatic disease patients. We know we don't have cure, but the next goal outside cure is longevity of life and improvement in quality of life. And this class of drug definitely confirmed to be able to do that. So based on this data, FDA approved it, and we now have access to this drug for our patients. Another thing I like about lutetium-177 dotatate is the simplicity of this drug and how to administer it. It's just 4 IV infusions, and each infusion is given every 2 months. So this is not something that patient has to take lifelong or several infusions, and there are decent spaces between each infusion. On a flip side, when this drug was initially launched, there was limited places which had capability to administer this type of radioactive drug. That has changed drastically. As I mentioned, there are over 200 sites in the US administering this. So access to this drug is a lot more easier now. And this is basically the first and the most important study that led to PRRT use in the United States in the NET patients. Now, let's switch gear. The topic of the talk is PRRT past and future. Before we go to the future, let's talk about the present. We discussed a little bit of the past. In last 2 years, we have had some exciting development building on the success of NETTER-1 already. NETTER-2 study was another randomized prospective study which tested lutetium-177 dotatate, but in the frontline setting. These are patients who are not being treated over over the years, and had not progressed on several lines of therapy. That was NETR-1. In this study, the investigator wanted to see what if we treat newly diagnosed patients or untreated patients, metastatic disease. However, these were more aggressive patients, grade 3 NET and higher grade 2, the Ki-67, more than 10%. The study design was pretty similar otherwise, 2:1 randomization. So 2/3 of the patient got randomized to get lutetium-177 dotatate, and 1/3 got higher dose octreotide LAR. And at progression, crossover was allowed. Dr. Simran Singh presented these data at GI ASCO, and now the data is already published and is open to public domain. Let's see the Kaplan-Meier curves here. Similar to NETO-1, this study was also very positive. The top line depicts the efficacy of lutetium-177 dotatate, and the bottom line is the control arm with higher dose sandostatin. So what do we find here? The median PFS in the study arm with the Lutathera in the higher grade 2 and grade 3 Velfenid was 22 months as compared to 8.5 months. Significant improvement in the frontline setting. So what does this study do? This study allows us to use Lutathera upfront, especially in more aggressive diseases, patients who have rapidly growing neuroendocrine tumor, and especially the grade 3 and higher grade 2 tumor. So this opens up the doors for use of that in the frontline setting. That doesn't mean that every patient should get treated with Lutathera upfront. We have to individualize the treatments. If patient has Ki-67 12% but is behaving in a very indolent manner like a grade 1, you may not want to use Lutathera upfront, but it does provide us access to use it in the frontline setting in the appropriate patient. So that's why it's a very important study, perhaps practice changing. Another important aspect of this study is the effectiveness of this drug in shrinking the tumor. And what we found here is that patients who received Lutathera reported almost close to 40% response rate. Okay, that means almost 40% patients showed partial response or disease shrinkage, which is not true for NETR-1. So the low-grade NET patients, this drug is mostly cytostatic. That means stabilizes cancer. Only 15-18% patients show shrinkage. Here, about 40% of patients shows disease shrinkage. That tells us that the higher grade tumor, the more aggressive biology tumor, is also more radiosensitive, and it shrinks a lot. So what does that mean for us in clinics? If a patient needs cytoreduction, that means it needs disease shrinkage, it's very symptomatic from large bulky disease, Lutathera may be a very good drug to shrink those, especially in the grade 3 and the higher grade 2 disease. So this was a very impressive looking response rate, in my opinion, as compared to other drugs we use in neuroendocrine tumor space. All right. And even more recently, earlier this year, ITM, another company, they revealed their data on a large randomized phase 3 study called COMPETE study. And this is the first time we have another competition to Lutathera. It's not FDA approved yet, but it's a similar type of drug. It's a lutetium-177 edotriotide. So same somatostatin analog linked to a radiopharmaceutical with lutetium-177, targeting neuroendocrine tumors, which has somatostatin receptors. Very same concept. It's just the somatostatin analog is somewhat different, so it is a different drug in that sense. But it is good for us in the field, for the patients, for the doctors, that we have multiple options. This drug is currently being reviewed by the FDA, and we hope that it gets approved, so we have multiple choices to offer to our patients. The study design was half the patients got the lutetium-177 edotriotide, the other half got comparator arm was everolimus, Afinitor. And the subtle differences between this and the Lutathera is 4 cycles, but each cycle was 3 months apart, unlike 2 months apart, what we are used to in the Lutathera arm. Okay? And what we found here is these are the Kaplan-Meier curves. These data was presented by Dr. Hume Capdevila at eNets earlier this year. And as you can see here, there was a statistically significant improvement in the median PFS, 23 months versus 14 months in the control arm of Afinitor. So a positive study, statistically positive, and we are awaiting whether this drug will be approved so that we have multiple choices. Choices are always good for our patients and doctors. So happy about this, another advancement in field of theranosis PRRT in neuroendocrine tumors. All right, so this is the current standard of care, right? PRRT is now mainstream. It's no longer experimental. We already have one FDA-approved drug called Lutathera, and we have other drug which has been proven positive in studies, and we are awaiting its approval, if it gets approved or not. But what is next on the horizon? Should we be happy with what we have, or can we improve on the current PRRT drugs? Can we make them even better in terms of shrinkage? Can we make them better in terms of longevity of effectiveness? The median PFS you heard in the 2 studies was 24 months and 28 months. But I want my patients to live longer. I want my patients to have PFS double that. So I think, and many of other colleagues in our field, they feel that this is a great start, but this is not the end. And we need to continue to challenge the limits of science and improve the effectiveness of these PRRT drugs and make them better and better. So good news, friends, there is not 1, 2, but several studies trying to improve on the first generation of PRRT studies. And the next segment will basically touch briefly on that, what's up and coming. So in PRRT, most of the research is happening either in alpha PRRT or beta PRRT. Alpha means it's a type of radiation, ionizing radiation. So different radionuclides emit different type of radiations. You may have heard of alpha, beta, gamma. It doesn't mean alpha is better than beta or beta is better than gamma. No, these are just these are physical types of radiations. They have different energies, wavelengths, and physical properties. So physics defines them alpha, beta, and gamma. Though there are some drugs that belong to the alpha category, that means they emit alpha ionizing radiation, lead-212, actinium-225. And then there are others which are beta PRRT, like lutetium-177. Okay, so a couple of slides on alpha. This is a slide that was shown by Dr. Delpasan a few years back at several conferences, NANETS, eNets, and others, and now it's in public domain. So they were testing at that time in phase 1 study a drug called lead-212 dotamtate. And you see patient here on your left-hand side with somatosensory receptor positive tumor and almost actually complete response after 4 doses of this drug. So very, very exciting and kind of caught the field by surprise. And of course, everybody was super happy. Since then, several years have passed. The phase 1 completed successfully and drug entered into phase 2. And then phase 2 is completed, but the data is continuing to mature and being followed up. Dr. Strasberg earlier this year presented some follow-up data as well as safety data and some efficacy data from a group or a sub-cohort at ASCO earlier this year. And basically what Dr. Strasberg presented is one of the cohort where the data was mature enough that cohort 1, 17 out of 36 patients had about 47% response rate. We still don't have the median PFS because the study is still not mature. They're still following up. Very, very positive and promising signs. But what we have heard, what I've heard personally from different conferences, the side effect profile for alpha and beta are somewhat different. So they are not identical drugs. And in future, there might be a role for both. That's what I envision. I think if we have a drug which is approved in alpha category and beta, they probably will both find home in the patient. We will have to figure out the sequencing. We'll have to figure out which drug might be better at what time. But this is definitely very promising advance in field of neuroendocrine oncology. The other very important study, which is ongoing right now at several places in the US and abroad, is ACTION-1 randomized phase 3 study. So what is this study? A very important study. So patients who have got Lutathera and have done really well, and now 2 years later, the cancer is progressing. They may want to try another PRRT agent because they had such a pleasant experience. So ACTION-1 study is giving those patients opportunity for a retrial of PRRT, but in this case, they're using an alpha PRRT, the actinium-based product. And the patients are randomized into whether getting the alpha PRRT versus the standard of care. That could be Afinitor, Sutent, or other medications based on the individual country they are at. So this study is enrolling pretty well. It's now in the phase 3 stage, and hopefully next few years we will see how the study rolls out. And if this is a positive study, it might lead to another approval after Lutathera. So very, very important study, and I've kind of highlighted the clinicaltrials.gov identifier. If this is something of interest to you, there are several sites in the US where the study is open. Okay. All right, let's switch gears in the last 5 minutes. What's happening with the beta PRRT? We discussed a couple of very important studies in the alpha PRRT. There is a lot of excitement going in the beta PRRT. So the first generation of beta PRRT, the NETR1, NETR2, the lutetium dotatate, were successful, very positive. And we are using, I've personally probably treated over 350 patients in my career since FDA approval. So hundreds and thousands of patients being treated with these drugs. Lutetium 177 edotriotide earlier in the year showed positive results. So this is good. But what's next for lutetium-based drugs? Well, we are combining them with various radiation sensitizers. These drugs have been combined with chemotherapy, with targeted therapies like PARP inhibitors, VEGF TKIs. These studies are all ongoing. Where I'm personally doing a lot of research in neuroendocrine oncology is in the field of radiation sensitizer. And I'm currently leading the triapine Lutathera study, as well as peposertib, which is a DNA PK inhibitor Lutathera combination nationally. So in today's talk, I will discuss a little bit on triapine because we have some data and some would be coming up in very near future. So what is triapine? This drug was originally discovered at Yale several years back. And what it does basically, it's stops a process within the cell, which helps us to make the building blocks of the DNA. Okay, so from ribonuclide to deoxyribonuclide, it stops that process, and this process is very important for DNA repair. So when a radiation-based drug acts on the cancer cell and damages its DNA, tries to kill it, why does the cancer cell survive? Why does it grow back? Because one of the rescue mechanism is that the DNA heals itself. The tumor DNA has mechanism to heal itself. What if we block that healing process? And the triapine drug does that, or at least tries to achieve that. We tested it in preclinical models, both in vitro and in vivo models. And we found when we added triapine, with the radiation, those tumor cells had significant reduction in growth as compared to when we did not add triapine. So these are higher doses of triapine, lesser growth. That's what it shows. Then we moved this drug from our lab into a first-in-human phase 1 clinical trial, and about 6 sites in the country treated about 32 patients. And the study was successful in the sense that we were able to confirm that we can safely combine triapine, which is a pill that patients take for 2 weeks with each Lutathera infusion, They were able to successfully take it, complete the course, and had no major side effects. We were able to find the right dose. And we also found very promising effectiveness based on the interim data. The government gave us a very big grant in form of a career development award, and I was able to take this phase 1 into a large randomized phase 2, which is almost a 95, 98 patient trial, currently enrolling in about 16 hospitals or universities throughout the US. The trial is actually almost done. Another month or so, we would be closing it for further enrollment. And this trial, half of the patients are getting Lutathera, other half are getting Lutathera plus this triapine pill. And when this trial reads out, we will know for sure adding triapine pill, does it improve the effectiveness of Lutathera or not? But later this year, we now have a matured data from the phase 1 trial that I initially described, because we now have followed some of those patients for good 5 to 6 years. We now know how those patients did who got triapine and Lutathera, and it looks very exciting. I'm not at, I'm not allowed to disclose the results yet, but we will be presenting these results in upcoming ESMO meeting in Europe in October. After that, I'll be very happy to discuss those with you guys at next NCAN. But let me just say, it looks very exciting. All right, last but not the least, last 2 minutes. So patients who've received Lutathera and done really well, as I mentioned, some patients go almost 3 or 4 years before the tumor starts growing again. And it's a natural question when the cancer starts growing again, the patient would ask, doc, that drug worked really well, 4 doses, and I had a good disease control for so long. Can I try it again? And unfortunately, the FDA has only approved it for one time, four doses one time. In Europe, they've been retreating for several years, and we've kind of done a review. Dr. Garima Gupta and myself and some other colleagues, we will be presenting some of that data at this year's NANETS. But we have done this review, and we have found that the drug is very effective, even if you retreat it, as well as it's also safe. But there's no prospective data to confirm that. So NCI sponsored this study. It's a joint venture between US and Canada. Dr. Simran Singh from Canada and myself are co-leading this study. And this study is now open in several places throughout the US. And what we are trying to do here is we're trying to retreat patients, and the name of the study is also net retreat, with additional Lutathera doses. And the key advantage here is that we don't really need insurance support to get the additional Lutathera. The Lutathera is being sponsored by Novartis, so we have easy access to drug, because the second time Lutathera may or may not be covered by insurance. So clinical trials is a very good way to get access to the care. But more importantly, it will give us very conclusive and clean prospective data in support of should we retreat anybody with Lutathera or not. So this study is ongoing, and anybody who may be a good candidate, please ask us, or you might find a site close to you that may be able to get you enrolled in this study. So very excited study. All right, with that, I would wrap it up, and so that we have some time for Q&A. Again, thank you so much, NCAN, for this wonderful opportunity.